Okay, good morning, honorable members of the judging panel, lecturers, and dear authors. Today, under my study, I am going to focus on evaluating the relationship between interferon gamma levels and anaplasmosis infection in sheep using ELIZA. So first, ovine anaplasmosis. Ovine anaplasmosis is simply a tick-borne disease which is caused by intra-erythrocytic bacteria, which is under the order Rickettsialis, family Anaplasmataceae, and genus Anaplasma. So this infection is primarily transmitted by ixovidae ticks. Apart from that, this can also be contaminated through contaminated surgical tools and equipment that may be used in surgical processes like dehorning. Currently, this anaplasmosis has become an economically significant concern as it's leading to jaundice, anemia, weight loss, loss of productivity, and even in severe cases, the death of the farm animals. So because of this reason, this has become a key concern not only in animal health but also in farm management. In the present table, there are 8 anaplasma subspecies detected. But according to the literature and under my study, I am focusing on Anaplasma ovis, Anaplasma centrale, and Anaplasma marginale. If I talk about this interferon gamma, it is a cytokine, simply a signaling molecule in the immune system which is responsible on activating the macrophages that helps to kill these intracellular pathogens, including these Anaplasma bacteria. So as these bacteria transmit through these ticks and as it persists in the red blood cells, it makes the eradication process difficult. So because of this role, it makes interferon gamma a key candidate biomarker in this infectious disease research as its level reflects the immune activation. So according to the literature and past studies, it shows the cytokine level, specifically this interferon gamma level, it may change over time. So this raises an important question. Can we use this interferon gamma concentration in order to distinguish whether an animal is infected or not? Yes, according to the studies, it shows establishing such a relationship is important for many reasons. One being, it is helpful for us to understand how the immune system of sheep react to this infection. Also, it shows the immunological pattern by analyzing this interferon gamma which is linked with this infection. Also, it has the potential to act as diagnostic biomarker in order to take more accurate and reliable readings of this detection in this infection. And also, it enables monitoring of the disease progression over time by tracking the immune response of this disease infection at different stages. Not only that, it also helps us in developing vaccines because knowing how this immune system responds is very much critical when developing an effective vaccination. So if I talk about my problem identification and justification, there was a significant number of death cases reported at Oramata NLDB farm in Anuradhapura among the extant population of red madras and locally crossed sheep. Interestingly, even though high mortality rate was observed, there was no visible clear clinical signs seen among the surviving animals, even though heavy tick infestation was seen among them. So my objective of the study was to assess this relationship between interferon gamma levels and the infection status of anaplasmosis in sheep. So under my methodology, all my samples were collected from the Oramata NLDB farm in Anuradhapura and all my laboratory testings were conducted at the Department of Veterinary Pathobiology, Faculty of Veterinary Medicine and Animal Sciences at the University of Peradeniya. So, under my evaluation method, I used ELIZA. And in this retrospective study, all the 30 blood samples had been already collected. And after conducting PCR, DNA extractions and sequencing testings, I was able to confirm the presence of ovine Anaplasma ovis and Anaplasma marginale. And out of those positive samples, I separated cereals and conducted these ELIZA testings using the ID stream ruminant interferon gamma kit by following the standard protocols. And after obtaining the assay results, I calculated the sample to positive value ratio, which is simply the SP value for each sample. And as I conducted this testing twice, I calculated the mean SP value for greater reliability. And finally, all those values were then used in the statistical analysis in order to explore this relationship between interferon gamma and the anaplasmosis infection status. So on the data analysis, my dependent variable was the interferon gamma concentration, while my independent variable being the anaplasmosis infection status, which was categorized as positive and negative. The significance level was declared as 0.05, and all these statistical analyses were conducted using the R software package 432. So these are the test results, test 1, the SP values I got for test 1 and test 2. And then I calculated the mean SP values, which I used in the data analysis. So in here I used 3 approaches. The first one is the ANOVA in order to compare the significant mean differences between the two groups, which helped me to analyze the immune response variation. And secondly, I used the correlation analysis to examine the strength and the direction of the linear relationship between the two groups, which helped me to identify the infection presence and the severity. And finally, I used the logistic regression to assess the relationship between two groups in order to identify how this interferon gamma changes with the infection. And after all this data analysis, I obtained a p-value of 0.3829, which was greater than 0.05. So as a conclusion, together with all this finding, I can say that there was no statistical significant correlation found between this anaplasmosis infection status and interferon gamma level, suggesting that interferon gamma is alone not a reliable biomarker in detecting this ovarian anaplasmosis in a small study group like mine, which is I only use 30 blood samples. So finally, as a recommendation, I would suggest on future studies to increase the sample size in order to obtain a more robust relationship between this anaplasmosis infection status and interferon gamma level, thereby enhancing the dynamic immune response of the sheep. And these are some of the key references that supported my research. And thank you very much.
Thank you for the nice presentation, Ms. Satyanarayana.